Hello everyone. Today I'd like to give you an overview of genomic sequencing or DNA sequencing. The term genome was introduced by the Hans Winkler in 1920 to denote the complete set of the chromosomal and extrachromosomal genes present in an organism, including a virus. Genome sequencing refers to the process of determining the order of the nucleotide bases adenine, guanine, cytosine, and thymine in a molecule of DNA or the genome of an organism. The methods of the sequencing have become a game changer in modern biological and medical fields. DNA sequencing. DNA sequencing is the process of working out the order of the building blocks or bases in a strand of DNA. Before we can sequence the DNA, it has to be cut up into smaller pieces that are inserted into plasmid DNA. This is a plasmid DNA, and then put into bacterial cells. This makes it possible to produce lots and lots of copies of it as the bacterial cells multiply. The DNA is then isolated from the bacteria and sent for sequencing. The isolated DNA is transferred to a plate where the sequencing reactions will take place. A mixture of ingredients is added. This includes free DNA bases like A, C, G, and T, and add a DNA polymerase enzyme, and after that the DNA primers. Modified DNA bases labeled with colored fluorescent tags are also added. These are called terminator bases. To start a sequence reactions, everything is heated to 96 degrees Celsius. This separates the DNA into two single strands. The temperature is then lowered to 50 degrees. This enables the DNA primers to bind to the plasmid DNA. The temperature is then increased to 60 degrees centigrade, and the enzyme's DNA polymerase binds to the primer DNA. DNA polymerase starts making a new strand of DNA by adding unlabeled DNA bases to the target DNA. It continues to add DNA bases until a terminator base is added. These terminator bases have been chemically altered so that no more bases can be added to the new strand of DNA. Once a terminator base is added, the DNA polymerase enzyme stops making DNA and falls away from the strand. Everything is then heated 96 degrees Celsius again to separate the new DNA strand from the original strand. The process of heating and cooling is repeated again and again to produce lots of fragments of DNA of different lengths. The length of each fragment depends on when a terminator base got added. The length of fragment depends when the terminator base got added. To read the sequence of the DNA, the various fragments are separated by length using a process called electrophoresis. A capillary tube is lowered into each well of the plate and the electrical charge is applied. This causes negatively charged DNA molecules to move through the capillary tube. Each capillary contains a porous gel. The shorter fragments of DNA move through the gel more easily than the longer DNA fragments. As a result, the fragments become arranged by size from the shortest to the longest. As the DNA fragments come to the end of the capillary, the laser makes the terminator base light up. The color is detected by the camera and recorded. Each terminator base is labeled with a different color. A fluorescent green, C blue, G yellow, and T red. The shortest DNA fragments will be first, and the longest read last. The sequencing machine record the color of the terminator bases as a series of the color blocks. Each color block represents the label terminator base at the end of each fragment of DNA. By covering the colors into the letters, we get the sequence of our piece of DNA. Thank you for watching of DNA sequencing or genomic sequencing. Thank you.